Topic name is DNA fingerprinting. So this is one of the important topic for NEET exam, and this topic from molecular basis of inheritance. So when it's come to the DNA fingerprinting, let us see what is the fingerprints. So fingerprints means a skin marks we can see on the fingers, a palm and sole. So these fingerprints are very much unique from person to person. The same way the DNA fingerprint also very much unique from person to person. So when I look at the entire human population, it is about 99.9% of human DNA absolutely same in the human population. How much it is all about? So 99.9% of DNA absolutely same in the human population. It is all about differences in only 0.1% differences in person to person. That 0.1% of differences that makes a person unique in a phenotypic appearance, right? So when you look at the human genome, it is all about 3 into 10 power 9 base pairs are there in the human genome. It is all about when it's come to the human genome or human DNA, it is all about, so 3 into 10 power 9 base pairs are there, approximately more than 3 billion nitrogen base pairs are there in the human DNA or human genome. It is all about what are the differences, it is all about only 0.1% differences. What is this 0.1% when it's come to the 3 into 10 power 9? It is all about 3 into 10 power 9 by 100 into 0.1. It is all about 3 into 10 power 6. So these many differences, this 10 power, so 3 into 10 power 6 base pairs differences are there in the between two different individuals. So if you need to find out the differences within the human beings to sequence entire human DNA, it is a very difficult process and it's a very expensive process. When you look at the human genome project, it takes more than 13 years of time and it takes $9 billion of money to sequence entire human DNA. So if you need to find out the difference between two different individuals or among the population, every time to sequence the entire DNA, it is a very difficult and expensive process. So with the help of DNA fingerprinting, we can compare two different DNAs, the difference between two different individuals with the help of DNA fingerprinting very easily and a very quick way. So with the help of DNA fingerprinting, we can find the difference between two individuals. The differences between two different individuals with the help of DNA fingerprinting. The DNA fingerprinting, the report it takes 3 to 4 days and it takes approximately 15,000 rupees for the DNA fingerprint report of any person. So it is a very quick way to compare the DNAs and to find out the uniqueness of someone's DNA with the help of DNA fingerprinting. So this technique invented by, there is a scientist from United Kingdom called Alex Jeffreys in 1984. So who invented this technique? So this technique invented by Alex Jeffreys of UK in 1984. To solve some parental issues, he invented this technique in 1984. In India, it is all about, there is something called a CCMB of Hyderabad, two scientists from Cellular Molecular Biology, that is all about Center for Cellular and Molecular Biology of Hyderabad. In 1986, Lanji Singh and KV Kashyap, so they used this technique first time in India to solve one criminal case. So now coming to the DNA fingerprinting, before going to the in detail of the technique, we need to understand certain points regarding human DNA. When you look at the human DNA, We know that as per the Human Genome Project, it is all about less than 2% genes. They are all only coding for. They are only coding part. Rest of all non-coding part. Means when you look at the human genome, the major part it is all about non-coding part, and there is a very minor part it is all about that DNA code for some proteins. Means what? It is all about coding genes. Means what? Each and every character. It can be a skin color, hair color, body color, whatever the character you take, that is controlled by one particular gene through a particular protein. That protein can be a hormone. That protein can be a anything that influence the phenotypic appearance. Now it is all about when you look at the human DNA, there is something called a bulk genome, and there is something called a junk DNA. So bulk genome it is all about coding part. Commonly it is all about. So bulk genome it is all about coding part. So that this genome that codes for a particular phenotypic appearance. Now coming to the junk DNA, this is all about non-coding part. This junk DNA commonly it is all about a non-coding part of the DNA. So they occupies a major portion of DNA and they are not coding for anything. They don't have any importance, but this non-coding part plays a very vital role in the this DNA fingerprinting part. So when it's come to the DNA, there is something called a repetitive DNA. Repetitive DNA means a small portion of DNA that repeats several times throughout the length of the DNA. That is what is called a repetitive DNA. So these repetitive DNAs are very commonly seen in the non-coding part. What is that repetitive DNA? A small portion of DNA means what? Let us see what is that. It means what? It is all about A, G, C, T. This A, G, C, T again repeats. So this is what is called. It is all about 4 nitrogen base pairs are repeating 3 times here. The same way, it can be 4, it can be 5, it can be more than 10 nitrogen base pairs. It can be any number. That particular portion repeats a certain number of times. That is what is called repetitive DNA. A small portion of DNA that repeats several times throughout the length of the DNA. That is what is called repetitive DNA. So this repetitive DNA very commonly seen very close to the satellite part of a chromosome. What is the point here? This repetitive DNA very commonly seen very close to the satellite part of the chromosome. That is the reason this DNA also called as, or this repetitive DNA also called as a satellite DNA. This is also called as what? A satellite DNA. So one more point we need to understand, what is the importance of this repetitive DNA, satellite DNA when it's come to the DNA fingerprinting. So when it's come to the any mutations which occurs in the coding part, that can immediately affect the phenotypic appearance. Yes, right? If mutations which occurs in the non-coding part is there is no importance for any particular character. If any mutations which occur in the non-coding part, there is no considerable effect on the phenotypic appearance. So the mutations which occurs in the non-coding part, in the junk DNA part, that plays a very vital role in the uniqueness of the DNA of one person or a particular person. So because of mutations in these particular regions of a satellite DNA, repetitive DNA or non-coding part, so that makes a uniqueness of a particular person's DNA. Means what? So one DNA in many forms because of mutations in the non-coding part, that is collectively called DNA polymorphism. One DNA many forms, that is what is called. So DNA polymorphism. Who is responsible for DNA polymorphism? It is all about mutations are responsible for DNA polymorphism. So this repetitive DNA, this satellite DNA, this DNA polymorphism, or this satellite DNA also sometimes called VNTR, a variable number of tandem repeats. What is variable number of tandem repeats? What is the difference between random and tandem? Let us say random means, let us say this is what the length of the DNA. There is something called A is somewhere, and again this A is somewhere else, and again it's one particular nitrogen base or some repetition randomly somewhere else in the DNA. That is what is called random. But when it comes to the first one, the first one, there is a some stretch of DNA that repeats these four repetitions. There is a that four nitrogen bases repeats several times throughout the length. It is not random. That is what is called a tandem repetition. A particular stretch of DNA that repeating several times throughout the length. That is what is called a tandem repetition. That is all about a variable number of tandem repeats. So these four are responsible for, or these four are a basis for DNA fingerprinting. These all are interconnected, all are very much similar. So this repetitive DNA, this satellite DNA, this VNTR, this DNA polymorphism, so that is the basis for DNA fingerprinting. Now, so what is the point is how we can separate this satellite DNA, how we can separate this repetitive DNA. Let us see. So this satellite DNA can be separated with the help of a density gradient centrifugation. What is that technique? So commonly centrifuges are very commonly is there in the laboratories. So with the help of a density, a gradient, a centrifugation, we can separate this satellite DNA. So with the help of this technique, we can separate this satellite DNA. When I look at the density gradient centrifugation, there are some major peaks are there are some minor peaks are there. So in this technique, there are some major peaks are there are some minor peaks. This is very important for NEET exam. So major peaks, so where we can separate the bulk genome, that is what is called coding part. In minor peaks, it is all about satellite DNA can be separated. So in minor peaks, it is all about satellite DNA can be separated with the help of a density gradient centrifugation. So before going to the discussion of this particular technique, we need to understand something. So with the help of DNA fingerprinting, we can only compare the unique regions of two different individuals or a particular person's DNA. So with the help of DNA fingerprinting, we can't sequence even a single nitrogen base. That is very important for example. We can't sequence anything with the help of DNA fingerprinting. Only we will get a report which is in a just like a barcode. There are different lengths in the thickness of that particular bar. So here it is all about, that point is very important for exam. We can only compare two different individual DNAs with the help of this technique. We can't sequence anything here, right? So now coming to the technique, now coming to the DNA fingerprinting technique. So before going to the technique, we need to understand 2-3 terms. The first term is very important. It is all about restriction endonucleases. These are very common. Restriction endonuclease enzymes. These enzymes are very commonly used to break down the DNA. So these enzymes, they can break down the DNA at a specific region. Restriction endonucleases are very important for your NEET exam. So restriction endonuclease enzymes are very commonly used to break down the DNA at a specific region. This point is very important for your NEET exam. The second point, what is important? So what is a VNTR probe? So VNTR probe means, so these are a very, most of the times it is all about a single nitrogen base. It can be adenine, guanine, cytosine or thymine, a single nitrogen base that is labeled with the radioactive substances which are prepared in the laboratories. Is all about, it can be a G, a C, T, which are radioactive labeled, means radioactive substances are labeled with this particular nitrogen bases, which are prepared in the laboratory. Most of the times it is in the solution form. It is all about VNTR, a probe solution, right? So that is VNTR probe solution. Now coming to the technique, it is all about why, what is the importance of this technique. So this technique is very important, plays a very vital role in the forensic science, parental disputes, population diversities and genetic diversities, evolutionary biology, and biodiversity conservation. There are so many are there. These five are very important. For example, a forensic science, we know that there is a forensic team is there. So if any crime happens, it can be a bank robbery, it can be a killing of someone, it is all about any crime happens. These forensic people, they can find the what are the clues are there. The clues team can find, it can be a hair follicle, it can be a blood sample, or it can be a bone, or it can be any part of any cell of the human body, that can be used as a sample to get the DNA fingerprint report. So there is a need of the sample of a criminal is required to produce the DNA fingerprint. So the first point, it is all about sample collection. If crime happens, there is a most of the time there must be a there is a possibility of any hair follicle, most of the times can possible of a criminal hair follicle or blood. If any robbery happens or any crime scene happens, there is a possibility of blood or hair follicle or whatever it is, that can be a source for DNA fingerprinting. So when it's come to the parental disputes, they directly take the blood sample or a tissue from the both the parents and the child, right? So here the first point, it is all about a sample collection. The sample collection from a particular suspected person, from the crime location or the parents. So it is all about the first step in the DNA fingerprinting. It is all about isolation of DNA from the sample. The first step, it is all about isolation of DNA. That is what I said, right? From the sample or from the parent, we can take the cell and we can separate the DNA from the particular cell. Even a single cell can be enough for DNA fingerprinting. So with the help of different techniques, we can isolate, we can separate the DNA from the cell. That is the first step in the DNA fingerprinting. The second step, it is all about digestion or fragmentation. Digestion or fragmentation of DNA. So digestion or fragmentation of DNA with the help of restriction endonuclease enzymes, they can break down, they can make different fragments of the DNA. That is the second step. It is all about digestion or fragmentation. So with the help of restriction endonucleases. The third step, it is all about agarose gel electrophoresis. When you transfer these fragments into this particular machine, this agarose gel electrophoresis can separate the fragments of the DNA based on the size. It makes bundles based on the size. So means based on the size, these fragments can move into the different distance. So we can separate the DNA fragments with the help of gel electrophoresis. So with the help of gel electrophoresis, we can make a we can separate the DNA fragments based on the size. Now the fourth step, it is all about denaturing. So denaturing means what? When you look at the human DNA, it is all about a double stranded DNA. Two strands are there. Both the strands are combined with the help of a very weak hydrogen bonds. So here denaturing means what? To make double stranded DNA into a single stranded DNA. It is all about, so dsDNA into ssDNA, that is what is called a denaturation. So denaturation with the help of, when you heat the DNA or by using some alkaline chemicals, so we can make single strand separate from the double stranded DNA. Now the first step is all about a Southern blotting. What is Southern blotting means what? In this technique it is all about the single stranded DNA. So we'll move into the particular membrane. So with the help of Southern blotting, so we can move this ssDNA into a one particular membrane. That membrane it is all about, it can be a nylon sheet or nitrocellulose membrane. The same electrophoresis machine. So when you cover this ssDNA, with a nylon paper, and on the top of that will cover again with a towel. So this towel will absorb the water, and during that process what happens, this ssDNA will move into the nylon sheet through the capillary action. So here with the help of Southern blotting, we are transferring the single stranded DNA into the nylon sheet through Southern blotting. This technique is developed by MS Southern, right? Now it is all about the sixth technique. It is all about hybridization of DNA. So hybridization means what? So now it is all about, we know that VNTR probe solution, right? So here is the VNTR probe solution. So here it is all about, this is a VNTR probes. In this VNTR probe, adenine, guanine, cytosine, thymine, these are radioactive labeled VNTR probe solution. When you immerse this nylon sheet, in this particular VNTR probe solution, where this nylon sheet with the ssDNA, so what happens here? This nitrogen bases, they pair with the ssDNA. So complementary pairing can easily happens when this ssDNA that combine with the different nitrogen bases, that leads to production or development of hybrid dsDNA. Hybrid double stranded DNA can form. So when nylon sheet with ssDNA that immerse into the VNTR probe solution. So the last step it is all about autoradiography. So here autoradiography it is all about. So this hybrid dsDNA, in this process, hybrid dsDNA covered with a X-ray film. Hybrid dsDNA covered with a X-ray film. Now this X-ray film emits some light in the form of different bands. This X-ray film will emit a light in the different bands. So these bands are nothing but DNA fingerprinting report. So how they can emit light? Because of in the hybrid dsDNA it is all about one strand. It is all about that labeled with the radioactive substances, where we can get a report in the form of different bands. So that is all about DNA fingerprinting technique. Let us see one example. Let us say there is a crime happens, there is a bank robbery happens somewhere else. So the clues team or the forensic team, they found some hair follicles at the location of the crime scene. So with the help of hair follicles, they generated this report from the crime location. Let us say that. from the crime location sample, so they got this DNA fingerprint report of a sample which is available at the crime location. So when any robbery happens, there are some suspected persons must be there. So what they will do that? So they will take a report from different suspected people. Let us say there is a suspected A, suspected B. So if the DNA fingerprint of suspected A, it is something like that. If the suspected B DNA fingerprint report, the suspected B fingerprint report that is matching with the crime location sample, now we can understand the suspected B person, the suspected B was the criminal at that particular point of time. So the same way, the parental disputes also can be solved with the help of DNA fingerprinting. They will generate DNA fingerprinting from both the parents from who are fighting for the child, and they will take the DNA fingerprint report from the child and they will compare. So one point is very important. It is all about the children will get 50% of VNTRs from mother or 50% of VNTRs from father. So with the help of DNA fingerprint report or fingerprinting, we can easily solve parental disputes also. So this is how a DNA fingerprinting technique can be done here. So thank you.